All right, so our next speaker is Darcy Telenko. She's our Crop Disease Specialist. So on this, Darcy and I went out to actually a field in Pinney Purdue, and we did a video. So first we're gonna watch this video, and Darcy's gonna talk about some diseases. And so we'll get to see some close-up stuff there, and then we'll be available for questions. So type those questions in the chat, and we'll see if we get this started.
Hi, I'm Darcy Telenko, Extension Field Crop Pathologist at Purdue University. And today we're here at Pinney Purdue Ag Center to look at our field plots and talk about field crop diseases. So one of the biggest questions we're getting this season is about the foliar diseases in corn, especially southern rusting corn and tar spot. So here I'm standing in front of our research trials up at the Purdue Pinney Ag Center, where we're trying to evaluate different programs for managing tar spot. As for this field, the disease is just beginning to start. In many of our fields that have had a history of tar spot, particularly in 2018, those are some of the fields we're monitoring this year as the disease is starting to begin and click up through the canopy. So when you first start looking for tar spot, it's important to understand your field history. Did that field have a disease problem in it, either with tar spot, gray leaf spot, northern corn leaf blight? All of those diseases are going to overwinter in the debris. So when we're scouting for those diseases, it's pertinent to start looking in the lower canopy at those lower leaves. Is the disease there? Is it active? And some of those indications can tell you whether a well-timed fungicide would be necessary. Here in our plots, the crop is about R1. We're beginning to find a few lesions of tar spot. When you're looking at it on a leaf, and I'm just going to pick a leaf for this example, you look closely on it. And if you think you have tar spot, the first thing we're suggesting our growers to do is scratch at it, because many times these early leaf symptoms may be mimicked by insect frass. And if it is insect frass, if you scratch at it, it's going to come off. Versus if it is a tar spot stroma, that stroma is growing embedded in that leaf tissue and it won't scratch off. Here's an example, again, of a stroma of tar spot that we're finding here in our research site. As you can see, it's almost a needle in the haystack. We found one little stroma on hundreds of leaves we've looked at. But if you look at it, it's embedded in that tissue. It's almost raised, coming up a little bit if you look real closely with it, if you have a hand lens. So this disease is starting to slowly appear, depending on your field site. This is 2 leaves down from the ear leaf. So again, it is slowly moving up in the crop canopy. Again, if you always have questions on tar spot, you can send us a picture or a sample to the Purdue Disease Diagnostic Lab. But as we continue to scout for tar spot, the disease will eventually move up the canopy. We are starting to find some fields where we see a few lesions on the ear leaf and above. And the idea when we're trying to time a fungicide is we want to manage and protect that ear leaf and above to try to get the best yield out of the crop. Tar spot of corn has been one of those diseases we've been tracking here in Indiana since we had a big epidemic in 2018. We're standing here in front of our research plots where we're trying to evaluate fungicide efficacy and fungicide timing for managing tar spot. We have begun to see tar spot in northern Indiana. We are tracking it where it's active. Last year we confirmed 68 counties that we were able to find tar spot. Of those, the counties that I'm most worried about are those in northern Indiana where we have the environmental conditions that may be conducive for this disease. The tar spot really likes high moisture conditions, generally lower temperature. So around the lake effect, around Lake Michigan is where we're seeing the highest incidence of tar spot, and that's where we've seen the greatest yield impacts. We track the activity of tar spot using a IPM map as shown here. You can see that we have a number of counties that are colored yellow, which indicates that we have found active tar spot lesions within the crop canopy. Also, the areas also show that we have 68 counties. We did confirm tar spot, so that means there is potential for that pathogen to be present.
To determine the risk of a field to a disease, it's important to understand the disease triangle. There are 3 main factors that contribute to plant disease: the host, pathogen, and environment. First is a susceptible host planted in a field. That may be the specific plant species, corn, soybean, or wheat, or also whether a variety or hybrid that you selected is susceptible to that particular disease. In addition, it may be important to understand what growth stages the various diseases may appear. The second thing to consider is a virulent pathogen present. Many of our pathogens we monitor in Indiana do overwinter in the crop debris, so it's important to understand if the field has had a previous history. These may include such things as gray leaf spot, northern corn leaf blight, tar spot, and frog eye. In addition, some of our diseases may move in with weather systems through spore movement, such as southern corn rust. This disease does not overwinter in Indiana, but each year we monitor its movement from the south. But once we know whether a pathogen is present and a host, the big factor that we need to consider is the environment. Is there a favorable environment that's going to promote the disease to develop within their crop canopy? Each disease has its own set of temperature, moisture, and leaf wetness requirements for that disease to occur. So it's really important to understand what those requirements are and if the environmental conditions are going to be conducive for that disease to develop. For example, if it's hot and dry, many of our foliar fungal diseases will not become a problem during the cropping season. But if the environment should change and we get a lot of leaf wetness and moisture, then there's a greater risk for those diseases to develop.
So when we know that we have tar spot present, then the question is, are the environmental conditions favorable for the disease to continue to develop within that crop canopy? So you can see here we have, I think, 11 counties now, but it may increase by the time this video comes out. But again, we're tracking this map to determine where we know tar spot is active. And this will just give you an idea of where those conditions have been conducive to start seeing the disease. Those farms in those counties really should get out and look around their crop to see if they can find it. If you have questions, you can always send me an email or give me a call. A second disease that we're monitoring in corn is southern corn rust. This disease does not overwinter in Indiana. Every year it blows up from the south. This year we knew that there were some southern epidemics that occurred early. There was a hurricane system that moved and blew some of those spores up in June. And we have begun to detect southern rust in many of our fields across the state. Again, we have a map that tracks this that'll tell you where we have found southern rust. So we have had samples sent in all the way from southern Indiana all the way up to Joseph County in northern Indiana. So we are tracking this disease. Again, it's something else to watch for. If it does get a hold, there may require a fungicide spray. When we're looking to try to control any of these diseases, we have to think about the best timing for putting out that fungicide. In general, for gray leaf spot, northern corn leaf blight, a VTR1 application has been very effective in helping minimize and reduce the yield impacts of those diseases. For tar spot, we're still trying to determine that best window. We encourage many of our growers to hold on to their sprays because we want that fungicide there for the 2 to 3 weeks when it's active, when the disease is going to be kicking up in the canopy. So many of our growers, I had them hold off until about this week, where many are starting to spray, where the disease is starting to kick up in northern Indiana. But we don't know beyond R3 if a fungicide application will be effective. So it's important to understand the growth stage of your crop, how active that disease is in the lower canopy. Is it really moving up? Will a fungicide then be effective in trying to manage that disease? So if you're trying to make a decision on whether you want to put a fungicide out in either soybeans or corn, it is important to understand your disease risk for that field and the diseases you're trying to target. In corn, it may be northern corn leaf blight, gray leaf spot are the two major ones here in Indiana that we might want to put a fungicide out to control. Tar spot is a new disease that we're also monitoring for that, in addition to southern rust.
This is a diagram of trying to understand fungicide timing. So again, we talk about being out there and scouting for the various diseases. In corn, we need to be looking at the late vegetative stages. Is gray leaf starting in those lower canopies? Do we find frog eye leaf spot in soybeans at the R1, R2 stage? Are those diseases starting to be active in the field, and are they going to trigger the need for a fungicide? So yes, we see that diseases are active in those crop canopies. We do decide to put a fungicide out, generally in corn, again, the VTR1, where soybeans are 3. And that fungicide is providing residual protection from a 2 to 3 week window. The idea is we want to lay the fungicides out when we know the diseases are going to build up enough to impact yield. Maybe in another field, the weather conditions have been slightly different. We don't see diseases for a few weeks later. Therefore, a decision's made to put a fungicide out, but it's had a later application window. That field B may show residual protection later in the season, and sometimes that may show the difference between whether one field or another sees a buildup of disease at the end of the season. And finally, if diseases aren't active until the end of the season, corn has reached R5, soybeans R6, generally addition of fungicide at this point is not going to give you that return on investment for putting it out there. So the idea is we really want to understand when are the diseases active, and we want to put those fungicides out when we know we can manage the disease and protect that yield. And we don't want to go too early because we may run out, the fungicides may run out of steam when we need later protection at the end of the season. So again, it's really important to be out scouting and looking for those diseases to make that well-timed fungicide application.
As for diseases that are occurring, gray leaf spot has been pretty active, so if you've had a field with a history of the disease, it's in continuous corn, gray leaf spot may be moving up to that ear leaf this year in Indiana. So here's an example of a leaf where we could have multiple diseases occurring. So a little bit of gray leaf spot starting, then we have a tar spot stroma occurring, and then also down in the leaf tip, there is some rust pustules. Now to confirm if I have southern rust, I'm going to have to take this back to the lab and look under the microscope to look at those spores to differentiate between southern rust and common rust. As for soybeans, they're relatively quiet. Across the state where we've been scouting, we found a few lesions here and there for frog eye, but nothing that really has triggered much of a need for a fungicide spray, unless you have a soybean variety that's out there that's highly susceptible to frog eye. And the other thing, looking at tar spot, as the disease progresses, it may be important to put a fungicide spray out if the corn is before R3. Okay. So we're out here in our soybean research trials here at Purdue Pinney Ag Center. We have a couple different projects going on. The first, we're trying to look at white mold management and how the best fungicides for that and timing. So we have some trials out looking at how to manage white mold. As a reminder for controlling sclerotinia white mold, this disease, the spores are released about the time of soybean flowering, and that's when the infection occurs. So it's highly important to understand the growth stage of the soybean to make that timely fungicide application. A few other things we got going on include looking at sudden death syndrome and how do we manage that disease. We have a bunch of different seed treatments we're evaluating on their effectiveness to try to protect the soybean roots from being infected by the fusarium and causing the disease to appear later in the season. In addition, we also run fungicide trials trying to rate efficacy of the different fungicides, especially if we get new products for frog eye leaf control. So there's a few different programs we have going on trying to best manage our soybean diseases. Luckily this year, many of our diseases are relatively low in soybeans. We have found a few frog eye leaf spot lesions around the state, but generally nothing that requires a fungicide spray. I am starting to see in areas where you have a history of sudden death syndrome, those symptoms are starting to appear in the field. So it is important to get out and look at your fields and try to identify if you have any of these problems appearing. Some of these soil-borne problems, again, you need to understand what's going on this year to make management decisions for the future. Our program will continue to track various diseases across the state. We have sentinel plots at all the packs in Indiana, trying to monitor the different diseases. This includes monitoring for southern rust, tar spot, and the various other soybean diseases. You can look at our maps and try to see what we see active, and we'll try to keep you posted on if any of these diseases should be of concern on your farm.
All right, hopefully that worked out okay. Everyone be able to see. Now for, if you have any questions for Darcy, you can put them in the chat. So here's a question for you, Darcy. With I know you're doing herbicide, or not, the fungicide trials for the tar spot. Do you have any early results on efficacy for that or timing, or you're still waiting for data from this year?
So this year's data is just beginning to start collection. We do have 1 year of data under our belts from 2019. In general, from that data we collected, most of the fungicides we evaluated were effective in reducing tar spots. So there is a number of fungicides available. The one thing we did learn is timing is going to be important. And maybe, Phil, can I share my screen maybe? I can possibly pull something up if would be helpful.
Yeah.
Let me go see here. I'll pull something up real quick. So what we learned is looking at the disease epidemic from last year, we put out timings, most of our fungicide efficacy timings, and we're doing a multi-state coordinated effort. That VTR1 did work. It managed to keep tar spot low for about 3 to 4 weeks. But then at the end of the season, you could see where the disease is picking up. And so I was trying to see if I can share one of my slides here, if I can find it. Just give me a second. All right, I'll share my screen here real quick. All right. Okay, so can you see that screen?
Yes.
Okay, so this is just an example of one of our uniform fungicide trials from 2019. So you can see on the 13th of July is when we first found tar spot in Porter County. And this disease remained relatively low, and then all of a sudden, towards the end of the season, it tends to spike. So you can see here in the non-treated control, this gold line, disease was low. We were keeping, with the fungicide application made at the VTR1, we reduced the disease cycle. So you can see all the fungicides kept the disease even lower. But then as that 2 to 3 week window starts to run out, that's when you can start seeing that tar spot began to expand in those canopies that were treated. And so that's the one thing we're really questioning is, if we had gone out in R2, would have that helped protect a little longer in the season? And would we have seen the benefit of a little bit more yield benefit than what we saw from that VTR1 stage? So that's what we were trying to encourage many of our growers, just to hold on a second and see if that later the R2 application works. For this season, we're evaluating all sorts of timing applications. So we did start at V10 and we have the VTR1, and our site at Pinney is a little behind, which is we're trying to catch that later disease. So hopefully by the end of the season, we'll be able to tease out if any of these later applications would be effective. We're going to go all the way up to the R5, and then some of our programs do have a double hit of trying to manage that disease. Let me see if I can share here. The thing with our fungicides after this year one here in Indiana, where we had that well-timed, that VT application, we did see overall many of our trials, we could get a 17 to 20 bushel yield protection. So again, where the disease got really significantly high in the field, these fungicides will help protect it and they will protect the yield. We just don't know if there's even more yield to protect beyond that. So that's some of the questions we're trying to get at. I am collaborating with Wisconsin, Michigan, Illinois, Iowa, and we're trying to look at some of the modeling and using some of these weather conditions. I know Lyndon talked a little bit about irrigation, but we do know that water is going to drive this disease. And it may even push it through, and you may have seen this slide before, but I just want to show this is an example of where they had multiple small irrigations, and even though this field had a fungicide applied, the disease took off. So water is a strong driver of the disease, and so that's just something we really need to monitor, and I think that's why in northern Indiana, that's where we see the greatest effect of tar spot and the annual effect that it's had occurring. I don't know if that answered it. Do you have any other questions, Phil? Questions, comments? We are trying to track activity through the state. So my team in another couple weeks, if we start seeing more tar spot activity, is going to try to get across the state to see how widespread it is. We didn't really get to the southeast side of Indiana, so we're going to try to start there. I suspect we could find 1 or 2 stroma across most cornfields during the season, but again, it's when it starts moving up in that crop canopy and we see that significant blighting, that's where we're going to start seeing the yield losses and the reduced yield potential for that disease.
So do you have any, we'll move on to soybeans, because I know you've been looking at resistance. Any updates to frog eye leaf spot to fungicides? Any updates on that?
Yeah, so when I arrived here at Purdue, we knew that there were some counties we had confirmed strobilurin resistance, the QOIs. I have a graduate student working on that, and I guess I could share my screen again. She recently got back in the lab, so we could, let me see if I can find it, look at distributor and resistance. So last year we went around the state, collected samples, and of course frog eye last year was a little light, but we were able to collect samples in, I forget how, 9 counties, or I think 10 counties. And so she just recently finished up some of this work. And so we have continued to confirm that of the 10 counties, 9 we were found strobilurin resistance in that Cercospora sogena population. We were surprised to find that we do have one county that all the samples we sampled from that county did not have the resistant genes. So there still may be some populations out there that don't contain the QOI resistance yet. But again, we'll be searching and collecting frog eye this season to see if that holds true. This means that when we look at trying to manage frog eye in our soybeans, first, if you can identify a resistant variety, maybe rotating to a non-host like corn to try to reduce that inoculum within a field. But then when you're looking at your fungicide applications, it's going to be really important, and we suggested anyways to use a multi-modes of action. So using a fungicide with 2 modes of action, the strobulurins could be in there, but you need to make sure there's an additional group like a DMI or a thiophanate within those products in order to try to manage this, especially if we're dealing with fungicide-resistant populations. And I think that's about, and we continue to track southern rust in corn. So here's an updated map. So you can see Georgia and Alabama, Mississippi have been seeing a lot of it, and it blew up, but we got it all the way up to Wisconsin. Again, if you have questions, we're trying to map these diseases. So tar spot, southern rust, or any other disease of interest, please, if you have questions, send them. You can send them to the diagnostic lab. We do have Indiana corn marketing support for research that we're doing, and that includes free samples, submissions for corn diseases. So if you want to send us a sample, there's free of charge for farmers in Indiana. So again, we're trying to just monitor and track some of these diseases to develop some better ideas on how to manage them. With southern rust as well, generally, if you look at some of the data that the southern pathologists have done, fungicide applications up to R3 might be effective, but anything beyond that, they're not sure that they'll get the return on investment if the disease is just moving in at that point in time. So again, it's just important to understand the activity of the diseases and how it's moving up in the crop canopy. Phil, did you have any other questions? I don't see any.
Yeah, I don't think I haven't got any other ones. So okay. Thanks, Darcy, for the presentation, and I'm sure I'll send out your contact info if people want to reach out to you if they have other questions. So